so for the next question for revision set 1, this is on biological molecules. All right, so in case you don't know which topic they are from, okay, biological molecules, and you can also link that to your enzymes, knowledge on enzymes as well. All right, so enzymes are polymers of amino acids. So basically, you know that amino acids is the subunits of enzymes, aka proteins. So I want you to remember by now that all enzymes are actually proteins. Okay. Now, also, in case you're not aware, okay, another term that we can see how we describe proteins, they are also known as polypeptides. Why? Because when amino acids join together, they form the peptide bonds. All right, and a quick revision. Okay, amino acids. Okay, with another amino acids, peptide. Okay, glucose. With glucose. Okay, that's glycosidic. Bonds, right? Right, and then between, so for your lipids, okay, your triglyceride molecule, okay, if you don't remember, we have, okay, 1 glycerol, okay, and 3 fatty acid. That bond is the ester bond. All right, and then between nucleotides, okay, one nucleotide with another nucleotides, okay, that is phosphodiester bond. Okay, remember that. Okay, so that's revision for you. Please check that you know all of this. All right, back to the question. So we must draw the general structure of an amino acid. So I've drawn that for you. So what you need to do is you complete the diagram. Okay, this is where the amino acid group is. Okay, so revision. This is the amino group. All right. Okay, and this is your carboxylic acid group. Okay, COOH, cool, right? Okay, COOH, like I use in class, cool group. So that's the acid group, and thus the name amino acid. Amino acid. Okay, and of course, what differentiates one amino acid to one another is the R groups. All right. So the R group. So if you got that correct, that will give you all the marks. Okay, although it's not necessary for you to memorize the structures of any particular amino acid, but what would be useful for you if you could remember at least get the smallest amino acid. Okay, structure, which is glycine. Okay. Okay, actually let me correct that spelling. So glycine. Okay, the R group for glycine is actually just, the R group is just hydrogen. Okay, so if we were to replace this, okay, what will happen in a glycine molecule? So glycine will have amino group, carboxylic acid group, but in the R group here would have been hydrogen. So this will become a glycine molecule. So this is the smallest amino acid. Okay, why is this relevant in our syllabus? Because I want you to remember that we learned about glycine and also lysine when we look at the structure of another biological molecule, which is in collagen. Okay, collagen. All right. Okay, let's get on with the questions. So when bananas are peeled, exposed tissue gradually turns brown in the presence of oxygen. Okay, this enzyme is due to catechol oxidase, which acts on the substrate catechol. So we know that that's the substrate for catechol oxidase. And then what happens is that with the presence of oxidase, okay, it causes banana to become brown because it causes the production of melanin. So the student investigated how the concentration of catechol oxidase affects the rate of this reaction while keeping all the other variables constant. All right, now a good revision would be, okay, having the knowledge at the back of your head. What are the other variables that should be kept constant, useful for your paper 3 and also paper 5, especially your practical? So for example, if you haven't, okay, at least you know, oh, what other things that we should remain constant? Temperature, for example, right? pH, right? okay, can be kept constant. All right, so for each concentration catechol oxidase used, student mixed the catechol oxidase solution with catechol and recorded the time taken for it to reach the standard Brown solution. Now, standard means, okay, we should have a prepared, okay, sample where we can compare, so that, okay, our results, our observations are the same. All right. Then they give us the formula. Okay, the rate of reaction for each concentration is 1 divided by time to reach standard Brown in minutes. And then they ask us to calculate, okay, when it reaches 2 minutes and 40 seconds. So what you must do is you can convert that into minutes. That's equivalent to 2.5 minutes. And then 1 divided by 2.5. And that's how we arrive to the answer of 0.4 minutes. Okay, 0.4 rates per minute. Okay, so I think that's relatively straightforward, and then they already gave us the formula, which is nice. And then they give us the graph which shows the results of the investigations that they did. So whenever we get graphs, always look at the axis. Okay, always make sure you check the axis. So on the x-axis we have percentage concentration of catechol oxidase, and on the y-axis we have the rate of reaction. Okay, in terms of practical skills, okay, paper 3 and paper 5, so I hope by now I want you to recognize that this at the bottom, that would be the independent variable, okay, what we manipulate, what we change. And then on the y-axis, this would be the dependent variable, so the dV, okay, which is what we measure. Okay, so the question reads, state how the results shows that the substrate was in excess at all concentrations of oxidase tested, because what we can see from the result is that the line was straight and it does not plateau, okay, at high concentrations of catechol oxidase, meaning it continues to increase, okay, because what happens if the substrate was limiting, aka if substrate was limiting, or we can say if substrate is the limiting factor, okay, the typical graph that we can see or we expect is that it should plateau. Alright, because what happens is that as you increase the concentration of catechol, you still have a lot of substrate available at the initial reaction, so rate of reaction increases, but once there is no more substrate available, it will start to plateau, meaning further increase in the catechol oxidase enzyme concentration will not increase the rate of reaction further because there is no substrate available, or its substrate has already been used up. So that means at that stage here, okay, here, that means substrate is limiting, aka you can also describe that substrate is the limiting factor. And how do we know that is not the case? Because the graph keeps showing an increasing trend. That means substrate is no longer limiting, okay, reaction continues to increase. So that's how I wrote my answer, so please take note. Straight, does not plateau at high concentrations of catechol oxidase. Moving on with the next question, the student carry out further experiment to investigate how the concentration of catechol affects the initial rate of reaction. Okay, take note that initial means the reaction at the start, okay, of reaction. All other variables were kept constant throughout this investigation. Okay, pH, temperature, alright, okay, volume, okay, are kept constant. For example, again, just be ready, have some thoughts at the back of your head how this experiment is done, again, for paper 3 or paper 5 for your A2 level. Alright, and then we look at the graph showing the effect of varying concentration of catechol. Okay, that's our independent variable, okay, IV, on the initial rate of reaction. That's our dV, dependent variable, what we're measuring. So let's look at the graph. Okay, so from here, again, for every graph, always look at what the axes are showing. Okay, so we have catechol concentration in moles per dm cube on the x-axis, and we have the initial rate of reactions per minute on the y-axis. Okay, we can see that there is a trend. Okay, there is an increasing okay, trend, and then eventually it starts to reach its maximum where it starts to plateau here. Okay, so that's the trend that we can see. All right, let's look at the question, what it is asking. So they wanted us to explain the shape of the curve. So we're not going to be describing, okay? We're not required to describe, you just immediately explain what is currently happening. So I want you to observe the graph that I have there, and then I want to read the answers that I have written here for you. So I will do is I will try to tell you which parts of the graph that I am explaining. All right, so if you read here, the rate of reaction increases as the concentration of catechol increases because there is high concentrations of the active sites and the substrate available. Now this part of the explanation is to explain what currently is happening here. So please take note, all right? Okay, so it's showing us, okay, what's happening at this increasing part of the graph. Okay, that's what my explanation is for. Okay, so that's the first bit. Why? Why is that there is an increase in the initial rate of reaction? Because there is high successful collisions between what terms? Between the enzyme active site, can you improve that again? Active site, and the substrate molecules. Okay, and then the next key term, forming the enzyme substrate complex ESC until a certain point where the graph plateaus. So until a certain point meaning until this point when the graph starts to plateaus. So at this point, which is what I've written there, at this point, okay, all the active site, okay, all the active sites are fully occupied, and we can actually use the word saturated. That's actually accepted, right? Saturated. Therefore, at this stage of the graph, what explains why the initial rate of reaction no longer increases is because the enzyme concentration is the limiting factor, or the reaction is limited by enzyme concentration. Right? So I've explained which part of the graph my points are explaining. So this bit where it plateaus refers to the last bit here. Okay, so what you can do is, can you please take note, have a look, and again take note at which point of the graph are these explanations are for. Moving on, they want us to use figure 3.4 to calculate the Michaelis-Menten constant, the Km value, between the catechol oxidase and catechol. Now, if we see the word use figures, that means we must show on the graph how we work the calculations out. Okay, so again, what you need to do is follow this step. Step number 1. Okay, step number 1, find the Vmax. Okay, we go to the graph. Go find the Vmax, is the reaction, get the maximum initial rate of reaction, and we can find here it reads at 8.2. Okay, we've done step number 1. Step number 2, find half of the Vmax. So half of that Vmax, so 8.2 divided by 2 would be 4.2. So that's our half Vmax. And steps number 3, find the substrate concentration at that half Vmax. So we indicate it on the graph. So that's half Vmax, the substrate concentration. Draw with your ruler, move down. So you read it off, that is equivalent to 0.21 moles per dm cube. So that means that is the substrate concentration at which the reaction occurs at half its maximum speed, which is also the definition of what Km value is, aka what Michaelis-Menten constant is. So hopefully that's where it is. Now the common mistake is that students normally just find the half Vmax and just quote the half Vmax. They forgot that they need to use and then find the substrate concentration. So please avoid that mistake, and remember they've completed all the steps 1, 2, and 3. All right. And last but not least, okay, this next question giving us information that methyl catechol has a similar shape to catechol, which is the substrate of this enzyme catechol oxidase. Okay, the Km value using this methyl catechol as a substrate was found to be much lower than using catechol as the substrate when the reactions were carried under the same conditions. Now that's very, very important. All right, state what these Km values indicate about the relationship between enzyme and the two substrates. Now whenever we have Km value, which is the Michaelis-Menten constant, it is really talking about the enzyme affinity to what? To its substrate, right? So I want you to remember this rule. So the higher the Km value, the lower the affinity, or vice versa, the lower the Km value, the higher the affinity of the enzyme to its substrate. So in this case, because we know that the Km value using methyl catechol is lower, so that means that catechol oxidase has higher affinity to methyl catechol compared to its own substrate. Okay, so that's how I would answer that question. And again, make sure you're comfortable with these terms, enzyme affinity. Okay, what it means is that if an enzyme has a lower Michaelis-Menten constant, that means it needs lower concentration of substrate for reaction to reach half of its maximum speed. So it's actually very efficient. Now, why is it relevant? Especially if we want to compare drugs, right? Or compare between different enzymes, okay? Because we want to, for example, say cut costs, okay? Efficiency, right? This is what we can use the information on Km value can give us, and that's how we can use it. So again, please revise on this one. Okay, you can memorize either one of these points and take note.